looks almost like a flying slug there.
Exactly. Hovering away.
Wait, it's not a flying slug?
Looks like you're getting ready to roast it over the campfire.
They char just like marshmallows. You know?
Yeah.
Yeah. I remember the first time I posted a picture of a banana slug out here, I had a couple people from home ask me if it was dumb. Like if it was just like scat or some rabbit poop.
I think you said dumb at first.
Dumb, no.
Like, that's about the
They are
intelligence of
kind of
banana
dumb.
slug. Yeah, like, well, there we start. We'll go ahead and get rolling. Oh boy, I've never clipped these onto these pants before. Put it in the back pocket. Here we go. Hello, thank you for coming to Brandon Bauer's thesis defense. And before I let him just go after it, I'm going to tell you a little bit about Brandon from my point of view and give you a little sense of who he is a bit more before we, get to letting him tell you about what he studied this entire time, which was not banana slugs. So one of the things that has really impressed me a lot about Brandon, especially all the way through from the beginning, like the first days that he stepped onto this campus until now, is really the independence, the ability for him to like just really tackle his project and go in directions that I've never gone before, but also do it in a way that really was able to do it without a lot of hands-on support and able to do a lot of interesting things. So in addition to that, I don't know if I've had many other graduate students, I've had as many deep philosophical conversations about really strange esoteric things as I have with Brandon over the course of his time here. Another thing, and this is really important with me, and this is true of all of my graduate students. I mean, many of these things are true of all of my grad students to some degree, but this thing really stood out to me is he also has a deep care for the animals that he works with. And this was exemplified last year at some point during the entire process of coming together with his thesis proposal. It's actually after we had this built that he came to me and he wanted to discuss, and we had a great discussion about whether or not spinning the octopuses and making them dizzy was going to be too, I don't know quite what the word, invasive to the octopus or damaging to the octopus. And we talked a lot about that. And I've had a lot of students I've worked with that have been really cavalier, not my graduate students, but I've had a lot of students that have been really cavalier with the animals that we've worked with. And Brandon has been the complete opposite of that, and I've really appreciated that. But also, and one of the big things overall that has been a huge thing is he has a very, very deep curiosity, and a kind of curiosity that doesn't just lead him to think a lot about things, but leads him to act and do something about the things that he's curious about. And this was kind of exemplified in that, so he took on kind of as a side approach to, or a side of some of the research that he was doing, he was interested in the movement of animals. This is something like how animals perceive their world, how they move about in their world has been something that has been interesting to him since I first started talking with him. And one of the side projects that he did was looking at, he wanted to look at how crabs move in a tank, but getting at that, he was going to simulate that, and in the course of that, learned a Python game engine and simulating how agents were moving inside some arena, kind of as a side interest thing that he was doing, and had little flocks of birds that were flying around in a simulated environment. I was very impressed with it. But really the point here being is that he has this curiosity that leads him to actually take on and do a lot of things that aren't necessarily required of him, or things that necessarily, I don't want to say not accomplish it, because they accomplish things, but still don't have like some immediate output to them, just for his own learning. So with that, I'm going to give the time over to Brandon, and I will turn that off. Okay, here you go.
Alrighty, thank you guys for coming to hear about my master's thesis. I focused this project mainly on octopus orientation, but a more specific way to put it would be that I was trying to see if we could use disorientation as a tool to actually look at the octopus's sensory systems and how those were working. And if you don't want to stay for the full 40 minutes, I can make it pretty simple for you. I took octopuses, spun them around, and saw if they got dizzy. But in order to actually understand why I was doing this, we need to back up a little bit and maybe start with something that we all kind of implicitly know, which is that we all use a wide variety of senses to kind of piecemeal put together a picture of the world, and specifically how the world relates to us. And this is true of us, anything as peripheral as heat coming from light through the window, or feeling the gust through a door, or even the drone of a presenter at the front of the room can really inform your understanding of the space that you're in. And this is no different for octopuses and crabs. This octopus is not only seeing with its eyes, but it's touching, tasting, and even sensing some amount of light through its suckers alone. And that gives it a good idea of what's in the tank and allows it to actually explore the tank. Now, similarly, this crab is trying to see, trying to touch and feel its way through the tank. It's just not nearly as good at building a model of what's around it. And so it's going to run right beneath that octopus. Now, as I said, all of these senses are important for understanding the world, but only some of these are more primarily important for when we're directing movement. And so when it comes to actually moving through the world, directing or coordinating movements, our vestibular sense, our proprioception, and our vision are a little bit more important. And so I want to go through those three senses to actually get a grasp on what these things allow us to sense about ourselves and the world and what that means for our orientation. So starting off, you may not be familiar with vestibular sense, but this is the sense of motion or of force. And so for us, that's located in our inner ear. We have 3 vestibular organs per side of our head. These are the utricle and saccule, which help us sense the force of gravity, as well as linear acceleration. Linear acceleration is changing your speed in one axis. And then we have our semicircular canals, which help us to sense angular acceleration. So this is how are we turning or spinning. And the sensation of our vestibular sense, it should be noted, is mainly focused on acceleration. So we're not actually sensing movement or raw movement itself with our vestibular sense. We're sensing changes in movement or changes in our velocity. And a good way to think about this is imagine you step onto an elevator and you're going to feel that initial lurch of the elevator as it starts to accelerate to its, we'll call it a cruising speed, but you're not going to sense too much movement once you're at that stable speed. And then you're going to start sensing that movement again when you decelerate once you're reaching your floor. And that'll be important for later, but I should note that with vestibular sense, because our organs are in our ears, we're only sensing force as it applies to our head. So to be able to build out a better picture of our body, we then need to add proprioception. And proprioception is basically the sense of where our body is and its general layout. So we have a wide diversity of mechanoreceptors, stretch receptors, and other things that are placed within our muscles and within our joints. And these receptors tell us how much our joints are bent, how much our muscles are contracted or relaxed, and they can even tell us if there's a stretch of our muscles, so if there's weight that's causing our muscles to stretch. And in one case, proprioception can essentially tell us what's happening to our body motion-wise. So we can sense through the skeletal layout of our body as well as the forces we're feeling from the stretch of our muscles. We can tell the direction of gravity, or we can tell the direction of forces acting on our body. But this also just gives us a skeletal layout that we can then connect to our head with our vestibular sense. So by combining these two things, we build a pretty cohesive picture of what forces are acting on our body and the higher clarity sense of force we're getting from our head, we can then apply to our skeletal understanding through proprioception. Now, we have an image of what our body is doing in the world, but we don't actually know what's going on in the world externally, and so we need other senses in order to complete that. And of course, there's things like hearing and other external senses, but when it comes to directing motion, for us, vision tends to be the most primary sense that we're using to actually move through the world and navigate through the world. And so then with all three of these, as I've been kind of leading to, we can integrate these three in our nerve centers, so our brain and our spinal cord and other places in our body, to come up with a picture of how we are relating to the world and how our motion is relating to the motion of the world. Now, our senses aren't perfect. We saw with that crab that it tried its best, but it just was not able to find that octopus. And so the accuracy of this sensory model we're building is extremely important to how well we're able to actually move through the world. And if we can naturally behave based off of the sensory system, or sensory model, if it's accurate enough that we can do the things that we would quote unquote naturally do, let's say walk in a straight line to the other side of the room, then we call this state orientation. But conversely, if that sensory model is inaccurate enough, we're sensing some motion that is in there, or etc., then that's going to make it harder for us to actually coordinate movements or direct movements in the world. And this is a state called disorientation. So disorientation is a dissonance between the sensory model we're building and how the world actually is. So that's how it works in humans. But why am I talking about octopuses? What is it about octopuses that allows me to compare and contrast humans and this much, much different animal? They are about 650 million years away from us in evolution. They don't have a skeleton. They have 8 arms that they can taste and see through. So what is it about them that allows me to kind of look at their disorientation in a similar conceptual framework? Well, the biggest thing is that they actually share 2 sensory systems that are fairly similar to us. So the first being their vestibular system. So octopuses have vestibular organs called the statocysts, and these are located in between their eyes. So I've marked those with white ovals on that left picture. And on the right is a diagram of the statocyst. And the organ that I'm actually talking about is only within this circle. It's actually hanging inside of a cartilage shell there in the octopus. And the statocyst is composed of 2 sensory systems. So the first sensory system senses gravity. And the way that it does this is there is a dense crystallized mass that moves in relation to gravity, and as it moves, it butts up against a wall of nerves, and those nerves essentially tell the octopus what's up and what's down. The second sensory component is in the posterior chamber of the statocyst, and this is just a chamber where the walls are lined with nerves, and there's a fluid that fills the chamber that, in accordance with the fluid's movement, the octopus can essentially decode with those nerves how it's turning or spinning. Now, that's almost exactly, like to a T, how humans are also sensing these things vestibularly. We and other mammals, and actually most other organisms, use dense crystallized masses to track the direction of gravity. And similarly, our semicircular canals use the movement of fluid called endolymph to track our angular acceleration. So these are very similar systems to our own. Now, similarly, octopuses have very similar visual systems. So on the left is an example of a vertebrate eye, vertebrate just being anything with a backbone, and on the right is an example of a cephalopod eye. So this includes octopuses, cuttlefish, and squids. And while there are functional differences, for example, we don't think that octopuses and many cephalopods can see color, at least not in the way that we can. Anatomically, it's actually pretty hard to find a difference. And of course, these are simplified versions of the eye, but anatomically they're pretty similar, and they use a very similar framework as to how to capture light and how to process that light. And so what we find is that these 2 sensory systems are very similar functionally to humans in octopuses, and conveniently these are also 2 of the main sensory systems that are important for coordinating movement and sensing movement for the octopus and for humans. So now the question is, how do we actually study disorientation? So I can kind of hypothesize here that maybe octopuses are going to be disoriented in the same way that humans are because of these similarities. So how do I actually find out about this? Well, one of the problems is we can't ask the octopus if it's disoriented. So when we're diagnosing humans with some sort of vestibular disorder, we can ask them subjectively, what are you feeling? What sort of disorientation are you experiencing? And obviously we can't do that for octopuses, so we need to find an external measurement that we can use to compare what we think is an oriented octopus to a disoriented octopus. And for me, this was movement. So I looked at what are the characteristics of their movement that I can track that might change when they're disoriented. But the problem with that is that octopuses are very strange creatures, and a lot of the time, even watching them all summer, I had a hard time even knowing if how they were behaving was natural, because they just move in such alien ways to how I would think to move. And so I wanted to solve the second problem by giving them something predictable to do. So I wanted to know what they were trying to do so I could better estimate whether they were doing it in a coordinated fashion or not. And so this is ultimately my solution to studying disorientation in octopuses, was to measure their movements while they went to catch a crab. Now the other big question about studying disorientation is how to actually elicit it. So I chose, as you know already, to spin the octopuses, but there's a reason for this, and we kind of have to look at why spinning disorients us. So if we go back to that elevator example where you're moving from a state of no motion to accelerating to a state of motion and then decelerating back to still, what you're actually sensing is a change in a state of motion. And so if we transplant that idea to a merry-go-round, what's going to happen is you're going to get on that merry-go-round, you're going to start spinning, and your body's going to be making a lot of little tweaks and adaptations to not only how you're processing the data that you're bringing in, the vestibular sensations and the visual sensations, but you're actually going to be even tweaking how your eyes are moving to bring in light, to adapt to this new state of motion. And it's not that new state of motion, that new state of spinning that you're sensing that's going to cause disorientation, but rather it's when that new state abruptly changes. So you're going to build this whole sensory model to adapt to this new state of spinning, and then the reality of your movement is going to quickly change to no motion. And all those little adaptations you made are going to carry over for a little too long. And that's what gives you these false senses of spinning or dizziness after you get off the merry-go-round. And so I assumed that because we're affected by that, we might also be affected, or octopuses may also be affected by spinning like that. And maybe unsurprisingly, no one had ever looked at this before. No one had ever thrown octopuses in a tank and spun them around. But there was a field of research back between the 50s and 70s where they were looking at octopus and other cephalopod visual and vestibular systems, trying to figure out how they worked. And a lot of this research was a little macabre. They were doing anything from partially blinding to fully blinding, surgically removing statuses, surgically attaching cuttlefish to metal bars so they could hold them in place. So just nothing that I wanted to be doing. I wanted to do this in a way where I could catch the octopuses, spin them around, and leave them back to the ocean happy and healthy. And the other important note here is that all of this research didn't answer the question as to whether octopuses showed or cephalopods showed any signs of disorientation from spinning or anything of the like. So, to take a quick breath, we've covered that disorientation is a dissonance between the sensory model you've built and how the world actually is, which can cause you to not be able to orient or coordinate your movements as well as you naturally would. We've also covered that octopuses have extremely similar visual and vestibular systems, and this is important because these are the systems that help us coordinate movement, detect movement, and even cause some of our disorientation from spinning. And then lastly, in order to study disorientation, we had to solve a couple problems, and the solutions to those problems were to measure the characteristics of octopuses' movements and to give them something predictable to do at the same time, which was give them a crab to go catch. So with all of that, I could start a thesis in which I wanted to solve three things. I wanted to determine if octopuses could be disoriented from spinning. I also wanted to assess the priority of visual and vestibular data. So if an octopus was getting conflicting data from its vestibular system versus its visual system, was one of these trumping the other? Was one of these held as more important than the other? And then lastly, I wanted to obviously establish an ethical methodology to do this, something where I didn't stress the octopuses out or harm them in any way. Now, the biggest and most immediate finding from this project is that octopuses are relentlessly stubborn, and they don't like putting up with behavioral projects, and so I quickly axed that second point. And this became a project about determining if octopuses become disoriented and figuring out how to do that in a safe and ethical way. So how did I actually do that? Well, I had a spinning tank built so I could actually spin the octopuses. Then I ran experimental trials with that spinning tank. And lastly, I made measurements off of the footage from those experimental trials to try and figure out if I was seeing a difference without spinning or with spinning. So I had that spinning tank built, and it consists of 2 drums. So there's the inner drum here, which is where the octopus would be, and so there's water, octopus in there, and then there's a red lid for that inner drum. And the point of the red lid is to essentially give the octopus a sense of comfort. So in the wild, octopuses are going to be hiding in dens, or they just like to be hidden or sheltered away. And so I was hoping that having something on top of the inner tank would be a little bit better than just leaving it open like this. And the point of it being red is that octopuses are understood to not be able to see red wavelengths. So if we filter all the light to be red, then when the octopus is looking at that screen, it's basically going to observe it as a black screen. But for us, we can still look through and see what's going on with the octopus. And so with the spinning tank, then I can start running trials, and they basically went like this. We would take the octopus from where we were holding it, move it to the experimental tank, put the red screen on for that shelter, and then they would get 1 minute of acclimation. And this was just for the octopus to take a breath, for it to de-stress if it was stressed out by being handled to get to the tank. And this is for 2 reasons. One, because I wanted the octopuses to be comfortable, but also because I didn't want any fear or stress to impact how they were actually moving during the parts we would be measuring. So then after that minute of acclimation, if it was a control trial, which means we're just trying to set a baseline for how octopuses naturally move, they would go straight from the minute of acclimation to the last step, which is we take the red screen off of the tank, we drop the crab in as far from the octopus as possible, and then we give the octopus a minute to catch the crab. But if it's a spinning trial, we go to, after the minute of acclimation, we go to a minute of spinning, and then they again go down, and after that spinning, we remove the red screen, we drop the crab in, and we give them a minute to catch the crab. Now, the tank was able to spin both drums either at the same time or separately, and it could also spin in either direction. But to keep things controlled, I only spun the inner drum. So I only spun the inner drum clockwise, and a good way to think about this is you're just sitting in an office chair and you spin around, so your surroundings, that outer tank is remaining still while the inner tank is moving with you. Now, I spun them at about 60 degrees per second, so to put this in context, that's every 6 seconds you're spinning 1 full rotation, so in a minute you're spinning 10 times around. And while I think that this would be enough to elicit disorientation because it's consistent enough, I also don't think that this was too fast to stress them out, and I can say that none of them inked or puked or showed any real signs of stress after being spun. So, ultimately, while it was on the slower side, I think it should have elicited a reaction if one was going to be there. Now, obviously, I needed octopuses to actually run the trial, so here are my 16 octopuses that I used for the experiment. These were collected by divers in the Puget Sound, and these are all ruby octopuses. These octopuses ranged from Crumb in the top right corner, who was just a little bit larger than a golf ball, to Dracula, who was about 10 times larger than Crumb and could easily pull my arm down into the tank with him. And so finally, I have the tank, I have the experiment set up, I have the octopuses, and I can record the trials. And what I need from the trials isn't the whole thing. I don't need the footage from all the steps. I just need the footage from that last step when the crab is dropped in, because I really only want to see what they're doing once they have a target to be going after. And so the measured parts of this experiment looked kind of like this. This clip would be where we're measuring the movement. And what I ended up with is about 200 trials, and those were at 60 frames per second. So you can imagine I have a whole lot of data. I have a whole lot of frames from these trials. And what I needed out of this footage is I needed the positions of the left and the right eye to actually measure the octopus's movements. So in order to label those 2 things, I wanted to avoid crazy amounts of man-hour in doing so. So I attempted to train a computer to do it for me. And ultimately what I ended up with is something that looks like this. So I showed a computer a bunch of videos of octopuses. I told it what the octopuses were, and attempted to make it label the eyes for me. And this is an example of the output I got from one of the tests with that computer, and the purple is showing the left eye, the red is showing the right eye, and those are all of the positions it plotted. Now on the left is the accompanying video that it actually processed, and what you should be seeing is you should be seeing consistent labels. You should be seeing consistent dots on the left and right eye, and you'll notice it's flickering a little bit. That third point we didn't use for calculations, but it is supposed to be there. But you'll notice it's not terrible, but we were missing big chunks of data, and it was even mislabeling the eyes at certain points. And this would have been just a headache to try and sift through, and it would have been almost more effort to go back through this data than to just do it myself. And so that's what I did, is I used my own biological neural network to go back through and hand label the left eye and right eye from every frame of every video. And I ended up with files that look like this, where I have the X and Y positions in the pictures of the left and right eye. And so finally, all of that, we can get to what I actually found and what I actually measured. So with those coordinates, I looked at quite a few different metrics about their movement and about their success in catching crabs. And these ranged from how fast they were moving, how long it took them to actually catch the crab, some directional things, so where they were facing and where they were moving to, and then even how often they were catching the crabs, so if they were more or less successful, and what types of movements they were using to catch the crabs. And the two most significant results that I found were a change in bearing heading difference and a change in the types of movements they were using to actually go catch the crabs. And you may not be familiar with bearing heading difference, and that's partially because I made that term up, but it's composed of two different directions that are actually used in the scientific field. Bearing, which refers to the way an object is facing. And so for my uses, bearing was where the octopus was facing, and I defined this by finding the position of the left and right eye, drawing a line between them, and then drawing a line perpendicular to that faced away from the mantle. So you can see this is the mantle. The bearing basically always faced directly away from that. Now that's where they're facing. The heading then is where they're moving. And so the heading I found by finding the center point of the eyes and tracking what direction it needed to go to get to the center point from the next frame. So I can do this for every frame, get a bearing and heading for every frame, and then pretty self-explanatory, I can find the bearing heading difference, which is just the angle between the bearing and the heading. And now the question is, why am I looking at that difference? What's important about that number? Well, if we think about how we coordinate these two things, so how we coordinate how we are facing and where we're moving, I would say on average, you're generally going to be moving almost in line with your bearing. We can move off to the side, but if you think about facing forward and moving to your periphery, it's a lot harder and less comfortable than just moving straight forward with your field of view where you have the highest visual acuity. And while octopuses may not share the same 0-degree difference angle bearing heading with the bearing that we've given them, they probably do have some sort of stable coordination between where they're facing and where they're moving that I think once disoriented would either increase or decrease or change in some way. So one way to think about this in humans is, let's say I'm trying to walk straight to the wall at the other side of the room. Likely if I was oriented, I would just walk straight there, but if I become dizzy, there's the chance that I'm going to stagger off to the left and right of that line, which means that the direction I'm moving is going to be further from the direction I'm facing. And so this increases the bearing heading difference. So I thought maybe if octopuses are disoriented, they're also going to stagger like that, because we're tracking the eyes, which is where their staticist is, it's where their eyes are. So this is going to be the point that they're controlling the most and may lose control of as they're disoriented. And so, as I said, we find bearing and heading for each frame. Then we find the angular difference, the bearing heading difference for each frame, and it's just that singular number that I'm going to be analyzing in the next couple frames, or the next couple slides. So, to analyze the bearing heading difference, I take all the frames that I have, so from when the crab is dropped in to when the crab is caught, and I put that in one data set called the full video data set. And I analyzed that by itself, but I also attempted to make this a little bit more specific by pulling subsets of data out of the full video that only included directed movements. And a directed movement is just a movement that ends in the catching of the crab. So this is just the movement that actually results in the octopus getting the crab. And these consisted of two different types of movement, either jetting or crawling. And it's going to be a lot easier for me to show you what these look like than to describe them. So these are a bunch of examples of jetting movements. Now, jets are very defined and coordinated movements. So octopuses are always kind of going to do the same thing. Generally, they're going to use their siphon, which is how they exhale water when they're breathing. They're going to use that siphon to basically exhale really hard, and that propels them through the water. And they're going to use that to kind of linearly launch themselves at a target. And you see that generally they're going to relax their arms to let them trail behind them, and they kind of just shoot at that crab. Crawling movements, on the other hand, are a lot less defined. This is more of a catch-all category because outside of jetting, the ways that octopuses move are extremely varied and hard to kind of throw into bins. And so generally, crawling movements are just defined as when the octopus was going to catch the crab and it just used its arms, it stayed in contact with the surface of the tank and then went over to catch the crab. And one thing you might notice about crawling movements is that they tend to pounce at the end, so they'll do this little splat once they get to the octopus. And this can kind of look like a jet, and the main difference that I kind of defined for these things is that they would kind of do this jetting motion once they had crawled to the octopus, but this is more of like you have your hand above the fly and you're just about to catch it. Whereas jetting movements were far more for traveling. So this was, they're getting to the crab before they catch it or to catch it. And so, as I said, I wanted to analyze the characteristics of both of these types of movement, but the problem was that octopuses just weren't willing to jet enough that I could actually do that for them. I could actually analyze that type of movement. And so I really only ended up looking at the full video data set and the crawling movements. And so now finally we can get to what I found about average bearing heading difference. And before I actually put the results on the screen, I kind of want to get our bearings for what we're looking at. So this is going to be the full video results. So this is every movement from when the crab is dropped in to when it's caught. And on the x-axis, you can see these are the degrees. So this is the number of degrees between the bearing and heading. And then on the y-axis, you have that's just telling you where the data is going to show up. So on the bottom of the graph, we're going to see the control results, and on the top of the graph, we're going to see the spun results. And real quick, before I put the results up, I want to make it clear that if we see a positive bearing heading difference, this means that the octopus is going to be moving to the left of where it was facing. Whereas if we see a negative bearing heading difference, this means the octopus is moving to the right of where it's facing. So now we can pull up the control results, and I'll take a second to kind of tell you what we're looking at. So each one of these singular dots represents the average from one trial. So this is the average bearing heading difference from one trial. We went in, took the bearing heading difference from every frame, summed them up, averaged them, and that was plotted here. And then from all of these averages, I then built a box plot, and the overall average is found as this dark black line. So the average here is about 60 degrees, and it's positive, which means they're generally left-leaning. So they're generally moving to the left of where they're facing. And we can look at that on a real octopus. This is a frame from one of the trials. And generally it's going to look like this. So in that video, the octopus is moving directly towards the crab, but you can see it's facing this way. And one important thing to note about that is that because it's moving more to the left, it's actually moving more in line with that left eye. And so as I described, generally when we move, we're going to have a bearing heading difference of 0 because we're moving in line with our highest visual acuity. Well, octopus's eyes face outwards, so we know that they have really wide fields of view, but we don't necessarily know whether they have binocular vision or if that vision is going to be strongest on the ends around the field of view of each eye. So it's kind of natural that we might see octopuses move in line more with one eye than the other. But what makes that interesting is that I found a pretty significantly positive result, meaning that they're generally leading with their left eye, because if they were using their left and right eye equally to move, this would mean that you would be getting a lot of negatives, a lot of positives, and those would average out to about 0. So we're seeing a predominantly left eye preference for when octopuses are moving. Now, what happens once we spin the octopuses? What's going to happen to how they're coordinating, where they're moving, and where they're facing? Well, if we see an increase in the amount of degrees, this would mean that, or in the average bearing heading difference, this would mean that octopuses are willing to move in directions that are further from where they're facing. Whereas if we see a decrease, this means that octopuses are more tightly coordinating where they're moving in relation to where they're facing. And this second one is actually what we ended up finding. So I found a significant decrease in their average bearing heading, where they're going from about 60 degrees to about 25 degrees between their bearing and heading. And I think that one possible explanation for this, if they were disoriented, is that if you're dizzy and you're trying to walk a straight line, if you're not too dizzy, you can kind of focus and make yourself walk in a straight line. You can really concentrate all your senses and walk steadily across the room. And the way that you do this is you coordinate where you're moving with where your senses are the highest. Now, this might have been happening for the octopuses too. They may have simply been trying to coordinate where they were moving and where they were facing a little bit more tightly so that they could move in a more coordinated way. But of course, we're looking at the full video right now. So this includes every movement in the video and not just the ones where they're going for the crab. So what do we see when we only look at crawling movements? So again, with the controls, we see that they have this strong 60-degree average. But unlike the full video, we see that after octopuses are spun, they actually end up not changing that average at all. So they actually aren't affected at all with their average bearing heading, or they aren't changing that significantly in any way. Now, this is pretty interesting for two reasons. One, I've already been kind of harping on, which is that it's likely, seeing that octopuses have this average of 60 degrees, 60 degrees, and 60 degrees, that they do prefer using their left eye. This is nowhere from conclusive, but it's like a strong suggestion. And I actually looked for other research looking at preferred eye use or preferred arm use, things like that, and found that Octopus vulgaris, which is the common octopus which is related to ruby octopuses, was found to have preferential eye use specifically when hunting. So specifically when they're targeting prey items, they were found to use a preferred eye. And specifically, it was also found that dependent on the individual, you would find a different preferred eye. So some common octopuses were left-eyed, some were right-eyed. And that could be true here too, because if some octopuses were right-eyed, it may have just gotten lost in the average. So it may have still averaged out left, just like if you averaged who was right-handed and who was left-handed for humans, most of us are right-handed, and so you would end up with a right-leaning average. The other interesting thing about these two graphs is that we found this significant decrease, but we didn't find it when we're only looking at them catching the crab. And the best explanation I can come up with for this is that maybe the disorientation that did occur with these octopuses was too brief to actually catch in the crawling movements. So the crawling movements by definition end the video because this is when the crab is caught. And so if disorientation was only going to be occurring in that first part of the video, it's not going to show up anywhere in this final movement. The octopuses may have simply waited to become reoriented or had enough time to become reoriented before they actually went to catch the crab. Whereas in the full video, that includes all of those movements throughout the entire video, and so this might be dragged down a little bit. So ultimately, what this data is telling us is that it's possible octopuses have a preferred eye. It's seeming to be their left eye, or ruby octopuses specifically. It's seeming to be their left eye, but it could be either. And also that decrease in bearing heading difference tells us that, one, they may have been more tightly coordinating their movement direction with their facing direction in order to maybe coordinate their movements better. And the fact that we didn't see it in the crawling movements means that maybe that disorientation was just too brief and it wouldn't have shown up in that final movement. Now, as I said, I didn't have enough data to actually look at jetting movements, and this is partially because octopuses just didn't want to do them after they were spun. So we found that in the control trials, we're seeing 76 crawling movements and 84 jetting movements to actually catch the octopuses. So this is about 53% of the movements that they used to catch crabs were jetting type. Whereas after being spun, it decreased all the way down to 13% of the movement. So it's a pretty decent proportional decrease. And I think there's two reasons for this, or two possible interpretations for this. One is that octopuses really were disoriented, and just like if you were dizzy and I told you to run to the other side of the room, you'd likely be more comfortable in walking than running or jumping. This is a pretty coordinated movement, jetting is, and so it's going to be a lot more comfortable for the octopus to stay in contact with the surface if they're disoriented and just move to the crab while catching its bearings. Now, the other possibility is that octopuses weren't disoriented, and rather they were simply unsure of what was going on. So I would say that if the room started spinning around us for a minute, you would also be kind of uncertain of whether that movement was going to start again, why it happened in the first place, what's going on in your environment, because this is a very unnatural thing for an octopus to experience. And so it's possible that octopuses simply just didn't want to be doing more vulnerable movement types like jetting when they could simply crawl or stay in contact with a surface while they went to catch this crab. And while both of these could be possible, I tend to lean towards this latter explanation. I think it's far more probable, and if we're going with like an Occam's razor approach, it's the simplest answer as to why octopuses aren't aren't jetting as much after being spun. And I think this also goes along with the rest of the results that I found. So I didn't really find all that many significant results. There wasn't all that much evidence that octopuses were changing how they were moving after being spun compared to before being spun. So they weren't changing how fast they were moving, they weren't changing how long it took them to catch the crab, and they even weren't changing other directional measurements like how often their bearing was changing and things like that. Similarly, they definitely weren't disoriented enough to not go and get the crab. We didn't find any difference there. And so the question is, again, why aren't we seeing more evidence of this? And obviously the biggest answer to that is maybe our conditions just weren't enough to elicit disorientation. Maybe they needed to be spun faster or for longer. But I will say that I don't think that that is the direction that this research should go. I don't think we should try harder to elicit disorientation, especially because I think there's a lot of ways we can look harder for it at the current conditions. And I've already mentioned a couple of those reasons. One being that maybe the disorientation is very brief. It's only right after they're done being spun. And if that's the case, maybe we just need to look harder at that very first part of the trial. Secondly, maybe the disorientation is too light. Maybe giving them something to do actually suppresses any evidence we would see of the disorientation because the disorientation is light enough that they can essentially kind of sober up and just walk straight forward. Also, I didn't have enough data to look at jetting type movements. And while it's possible that after being spun, octopuses just won't want to jet in the first place, if it's the case that we can get them to jet after being spun, I think that this would be a really powerful tool for looking at disorientation because, as I said, these are very linear and targeted motions. And so if anything was to be disrupted when these octopuses are dizzy, it's probably going to be this kind of flailing targeted launch. And so if we can find differences in that, I think that would be really good evidence that octopuses do experience some amount of disorientation after being spun. And then lastly, and this is a more loaded point, one solution to not finding evidence might be to, rather than looking at the directional change of averages, maybe we want to look at the spread or the variance of the data. So maybe octopuses don't, on average, move faster or slower, but maybe they're more willing to move in wider or smaller ranges of speeds. And maybe speed is a weird example for that, but our other metrics, I think that would maybe benefit from, even average bearing heading difference. It may be that the change in the size of the range they're willing to move in is more important than the average bearing heading difference. And so ultimately, I found it very surprising that octopuses showed almost no difference in how they were moving. Because even if these conditions don't elicit disorientation, you would still expect that this sort of unnatural whole environment spinning scenario would at least make them take more time to catch a crab or maybe move slower, but I guess they're a little too voracious for that. Also, though we did find results, we did find that they were decreasing their average bearing heading difference and that they were jetting less after being spun. I think our results were far from conclusive that the octopuses were actually being disoriented or that was due to disorientation. But I do want to say that I also don't think that our lack of results means that octopuses aren't being disoriented, and I think we can look a lot further into this type of data and in these conditions to maybe find more conclusive evidence as to whether this is happening or not. And then lastly, maybe unexpectedly for me, I did find solid evidence that octopuses may have preferential eye use. Of course, this is going to take more research and more conclusive evidence to say that this is happening, but it's at least an exciting start to some new understanding of how octopuses are moving. So before I end, I want to thank all the people that helped me get this from an idea to a finished thesis. So I want to thank my family and friends who are watching from around the country. I want to thank the OnThink Lab, which includes my partner who helped me wrangle the mischievous octopuses and carry a bunch of 5-gallon buckets while we changed the water for every trial. I want to thank my thesis committee, Dr. Kurt OnThink, Dr. CJ Brothers, and Dr. David Cowles, who helped me with various aspects of this project. The divers who helped catch the octopuses, some of which are here today. And I want to thank Walla Walla University support systems who actually built the tank that allowed me to do this experiment in the first place. And then lastly, and maybe most importantly, I want to thank the octopuses that let me care for them, handle them, watch them, and even spin them. Thank you.
So you had 16 octopuses with a 10-fold size range. Did you see any difference between individuals?
So we used a linear mixed effects model to look at whether we were seeing differences on average. Do you mean that were we seeing differences in like some octopuses moved at this speed and some moved at this speed, or were we seeing them change their movement more in one octopus than
Yeah,
the
you
other?
have like, for example, you have the, what was the vampire, the huge one.
Yeah, Dracula, yeah.
Did that one act differently than your tiniest one? Like it was more left eye dominant or less left eye dominant?
So I didn't actually look at that, so I can't answer questions about that, but yeah. Question, yeah.
Do you think that there's like a way that you could study disorientation that would be more like their natural environment, like getting caught in a wave or something like that?
So yeah, that was something I thought about when I was first like coming up with the idea for this, is when would they actually be experiencing some spinning like this? And with a wave, you're still getting like kind of just that one axis, but it would almost be that it's happening. I feel like generally an octopus in the water would be spun lengthwise rather than like sitting up. So that would be something that would be really interesting, and I think that would actually be more naturally applicable, but how to get them like that is a hard.
Like a tidal pool.
Yeah, tidal pool.
A big wave.
Just get a wave pool, yeah.
Go remind me why you settled on 10 revolutions per minute.
So that actually was a little bit more like by gut judgment, because I looked at a bunch of other papers and there were no studies that looked at spinning cephalopods, but there were studies that looked at spinning mammals, humans, and some other animals. And so I looked at the rates of speed for those animals, and I kind of went a little bit below that just because I don't know what octopuses would or wouldn't find comfortable, but looking at these other animals and the ones that like in the field where it's been tested a lot more and they understand that it's a little bit safer for them, they were spinning them closer to like 90 degrees per second. So it was really just, there was no prior setting for this, and I was just kind of making a guess at what would elicit disorientation without stressing them out.
And was there no prior for like other invertebrates or other aquatic?
Not that I think the closest I found was a turtle, was that they spun a turtle, or they were looking at the optic reflexes in turtles. So that was the closest that I found, but I didn't find any like true invertebrates. Yeah.
You spun them clockwise, right?
Correct, yeah.
Did you think about like would it elicit a different response or result if you spun them counterclockwise? Because it's like if you spin the room one way, then you stop, you see it, continue to go, and then you go the other way, and it goes the other way. That like would affect the way that you adjust your movement.
The leaning. Yeah, that actually was one of the questions that I had, but with the amount of noise that was in our data, because just movement in general is a very noisy thing and not every change in a metric actually means something, I just wasn't going to have enough trials to actually like statistically say anything if I spun them both ways. But yeah, that was something that Dr. Onthink and I talked about, and I think would have been pretty interesting. Yeah.
What the octopuses actually do when they were being spun.
S-
In particular, I was thinking, so you spun the tank, it went around 10 times.
Mhm.
Did the octopus actually go around 10 times, or were they
So-
some different number?
So it did depend on the octopus. Some results that I didn't have time to explain up here were, we did find one other significant result, which was that we found that in spun octopuses, one of their movement metrics, which was the amount of change in where they were looking, decreased over time, and only in the spun trials, which kind of points at maybe some acclimation to the experiment. I don't know how necessarily. But ultimately what I found is that at the beginning, octopuses were more likely to be kind of loose, and they would spin with the water in the tank. And then as the experiment went on, they tended to kind of lock down more so. And originally I wanted them to be perfectly in the center of the tank, which I was able to get them kind of generally in that area, but yeah, it kind of depended on the octopus, but the majority of the time, especially by the end, they were kind of locking down to the surface, which would mean that they were traveling at the rate of speed.
Okay, there's a couple online questions here. First off, from Adam Burns. All of your octopuses were from the same region. Has there been much research on whether region may cause differences in these octopuses? For example, jetting versus crawling, left, right eye.
I would say that there's a lot of things that could change dependent on region and especially population. We know in other animals, population can change behavior, and it can also change different aspects of your morphology or like your body. And so, I mean, with something like jetting or movement type, it's possible that octopuses that are catching faster moving prey might be more apt to jet because that's going to be a quicker or more ambushy tactic. As far as disorientation, I think it might be harder to answer that question. Maybe if there's octopuses that are coming into tidal pools to hunt, maybe they are more exposed to kind of those surf conditions that would be spinning them around or kind of opening them up to more disorientation or more involuntary motion. But other than that, I'm not exactly sure how your population or region that you're in would affect disorientation. Yeah.
There's another online one from Alan Verdi. Were the same octopuses used for control and spinning to do paired comparisons?
Yes. So yeah, we used the same octopuses for the control and spun trials, so it just depended on the day which trial they were getting. Yep.
This is kind of going back to Stefano's question about like the direction that they're spinning. For the controls and for the spun octopuses, did they? You might have already mentioned this, but did the controls also show like a preference for the left eye?
So yeah, the controls. So the controls also showed a preference for the left eye. So we know that that wasn't from the direction of spin from the trial, or at least we know that maybe that wasn't a preference in the eye, but we at least know that that result wasn't from spinning. Yeah.
Do you think that if they spun the opposite way, maybe they're like, because their difference went down, right?
Mm-hmm.
When they were spun clockwise.
Yeah.
If they're spun the opposite way, it might go like more, like a bigger.
Possibly, yeah. Especially
Could switch.
like possibly if they're not trying to move in the direction that's away from where they're facing. Yeah. No, I think that could lead to like a greater average bearing heading difference. Yeah. Yeah.
Maybe you did, but I'm not sure you answered Alex's question. You did the same octopus was both in some control experiments and in some spinning experiments.
Yes.
But he asked whether they were paired comparisons rather than just average for everything.
I understand. Yeah. So for simplicity and the ability to explain it up here, I was showing you average results. But what we actually did was we used a statistical model called a linear mixed effects model, and this essentially controls for what are called fixed and random effects. And so fixed effects are going to be like which individual octopus you're talking about, where random effects are going to be the changes in certain metrics and stuff. So the statistical model we used did correct for the fact that we were looking at the same octopuses, and it also like paid attention to the fact that we're looking at, let's say octopus A is moving at like 5 pixels per second and it changes to 10 pixels per second, but octopus B is moving at 10 pixels per second, it goes to 15 pixels per second. That's not going to be lost in an average. It's going to be compared because we know it's the same octopus. So yeah, we did look at we factored in the identity of the octopus into how we were actually analyzing the data.
Okay, any further questions?
Yeah.
This is kind of a dumb question, but on the last slide, there was an octopus named number 11, I think. And I just wanted to know, like, what the thought was into that.
So I named all the octopuses as we got them, and generally some of them would sit unnamed for a little bit because I was kind of figuring out like what type of octopus they were or what name fit them. And they all had little name tags, and number 11, I have the name tag, the piece of duct tape, but I don't have any name on it. And I remember thinking about the name for so long, and I cannot for the life of me remember if I named that octopus or not. So unfortunately, he was a little lost to time.
Okay. Any further? Okay, well done, Brent. Thank you. At this point, we'll take a small break, about 10 or 15 minutes, and here at about, let's say, 4:15, his committee and him, we will meet in the lounge, and